Hi everyone, welcome to my presentation. The title of our paper is Collagen 11 Alpha 1 Structure Prediction by AlphaFold 2. Workflow for this paper. There is no structurally determined protein information for the aminopropeptide domain of collagen 11 alpha 1. So we have built three structures based on two different templates. One is 1DYK, which is a part of laminin alpha 2 chain, and another one is 2UUR, which is the ancestral domain of collagen type 9. Two different programs were used for this. One is modeler, another one is AlphaFold 2. Modeler generates structure based on homology modeling, whereas AlphaFold 2 is a neural network based model. Our lab previously used 1GYK template and 2UUR template with modeler to predict the aminopropeptide domain of collagen 11 alpha 1 chain and carried out some in silico studies. In this study, we will use AlphaFold 2 to predict the structure and compare the three structures. We have carried out comparative structural analysis using molecular dynamics simulation, shortly known as MD simulation, to deduce the best structure in aqueous environment. The purpose of this work to obtain the best possible predicted structure of the aminopropeptide domain, shortly known as MPP, of collagen 11 alpha 1 chain, to characterize the protein's structural stability and evaluate its behavior in an aqueous environment, and to build the best possible predicted structure of the NPP domain for protein-protein interaction study and protein-ligand interaction study in the extracellular matrix, and evaluate its possible role in the fibrillogenesis. Before I dive into the main paper, I will briefly go over the composition and function of the extracellular matrix, shortly known as ECM. The ECM is a non-cellular, three-dimensional structure made of many cell-secreted macromolecules that provide structural and biochemical support to surrounding cells. Structurally, ECM can be briefly summarized as a blend of water, fibrous proteins, and polysaccharides like proteoglycans and glycosaminoglycans. Collagen is the main structural component of the ECM, which provides tensile strength to tissues, regular cell adhesion by anchoring to integrins, and trigger cell differentiation and survival. Besides, the collagen lattice interacts with non-collagenous glycoproteins like fibronectin, laminin, or elastin, favoring the reinforcement and spatial organization of the extracellular matrix. Fibronectin and laminin fibers play a pivotal role in ECM assembly, acting as adhesive proteins. Next, types of collagen and their distribution. Collagens can be categorized based on size, distribution, function, and structure. Based on these findings, they can be classified as fibril-forming, fibril-associated collagens with interrupted triple helices, network-forming, membrane-associated collagens with interrupted triple helices, microfibril-forming, anchoring, network-forming, multiplexing, transmembrane, and miscellaneous. Type 11 collagen is a minor fibrillar collagen and is found co-distributed with type 2 and type 5 collagen. Collagen type 11 is mainly expressed in the cartilage and intervertebral disc. The in vitro studies suggest that the regulatory behavior of collagen type 11 is dependent on the complex formation within the amino terminal domain, shortly known as NTD, of alpha bond chain, which are proteolytically removed. Although this removal is slower compared to the alpha 2 chain and alpha 3 chain of collagen type 11. Thus, its presence on the surface of the fibrils may impose a steric hindrance and limit further deposition of the collagen triple helices to the growing fibril. This may result in lateral growth restriction. However, at the same time, it may provide stabilizing interactions between collagen fibrils and with other extracellular matrix components. This slide shows a schematic of 10 plus 4 microfibril structure of a thin cartilage collagen fibril, a pair of collagen type 11 molecule. Collagen type 11 microfibrils compress half of the 4 microfibril in the core of the structure, surrounded by 10 microfibrils at the surface. Here, the blue represents type 2 collagen, yellow represents type 11 collagen type 11 molecules, and the red represents collagen type 9 molecules. There is no experimentally determined protein structural information for the NPP domain of collagen type 11 alpha 1 chain. However, the NC4 domain structure of collagen type 9 has been experimentally determined, and these domains are homologous. This domain is a member of laminin, neurexin, and sex hormone binding globulin, shortly known as LNS domain. The LNS domain may interact with heparin and heparan sulfate, which is present in several proteoglycans in the extracellular matrix. The NC4 domain of collagen type 9 interacts with heparan sulfate, and this domain is also exposed to the collagen surface. It has been hypothesized that heparan sulfate or similar glycosaminoglycans may fulfill the bridging function between adjacent collagen fibrils and the cell surface, which may explain the role of the LNS domain containing collagens in the process of fibrillogenesis. Perhaps it may also explain the apparent role of LNS domain in the matrix-matrix interaction, as well as the interaction between cells and their immediate environment. If this is true, then the LNS domain could play a central role in the tissue integrity. Here, the collagen 11 alpha 1 domain also aminopropeptide domain of collagen 11 shares some homology with the LNS domain. This is the schematic representation of the aminopropeptide domain of collagen 11 alpha 1 chain. This globular region is the aminopropeptide domain, and these regions are the variable regions. The dimensions were determined by transmission electron microscopy. Next, the three structures which are built by using two different programs, modeler and AlphaFold 2, and two different structures, 1DYK, which is the alpha 2 chain of laminin, and 2UUR, which is the NC4 domain of collagen type 9. The first one is the 1DYK template-based structure built by modeler. We have used version 10.3 of the modeler software. The second structure is built by modeler using the template 2UUR, which is the NC4 domain of collagen type 9. And the third structure is derived from AlphaFold 2. So after building the structure, we have aligned the three structures using the match and align function of Chimera, which is a molecular visualization software. Here, the green indicates the beta strands, The yellow indicates the alpha helices and rest indicates loops. The gaps are indicated by dot. Interestingly enough, the structure derived from AlphaFold 2 and 2UUR based modular structure shares the similarity in terms of like secondary structural information. This slide represents the confidence level score of the structure which is derived from AlphaFold 2. It was worth mentioning that two amino acid residues which form disulfide bonds cysteine 25 and cysteine 207 and cysteine 146 and cysteine 200, they have found in close proximity. So the disulfide bonds were introduced using chimera. Moreover, there were several salt bridges observed, and the attractive charges interactions have been identified between amino acids lysine 127 and aspartic acid 125, arginine 130 and aspartic acid 207, arginine 39 and aspartic acid 183, arginine 28, aspartic acid 135, and several other similar kind of salt bridges. AlphaFold 2 showed the highest accuracy compared to the other two structures. While we used the Ramachandran plot, we have found 0% questionable observation and approximately 98% highly preferred observation and 2% preferred observation. The PLDT scores of the AlphaFold 2 derived structure are presented in the next slide. However, previous circular dichroism spectral analysis showed that there are approximately 33% of beta sheets in the aminopropeptide domain of collagen 11 alpha 1. From the structure we have got from the AlphaFold 2, we have found that there are approximately 32.7% of beta sheets, which are very close to each other. Moreover, we have found total 12 anti-parallel beta sheets from the AlphaFold 2 derived structure and 3 small alpha helices and 3 310 alpha helices. The beta sheets formed a sandwich. It is well documented that the 310 alpha helices are dynamic. They are intrinsically less stable compared to the alpha helices, compared to the regular alpha helices. So this is the table. Here it shows that only 4.9% of the amino acid residues are in very low confidence level. Approximately 1.4% of the residues are in the low confidence level and 7.2% are in the confident level, and rest of the residues are in very high PLDT confident level. Next, we have used molecular dynamics simulation to get some structural information in an aqueous environment. Our first experiment was to see the root mean square deviation of the protein backbone, which indicates the conformational stability of the protein. Among the three structures, RMSD found from the AlphaFold 2 is the lowest, which is 0.23. And the highest was found from the 1GYK based template, which is 0.69 nanometer, and the template 2UUR gave us a average RMSD value of 0.35. Next, the residue specific fluctuation of a protein, which indicates the structural flexibility, the individual amino acid residues structural flexibility. From the AlphaFold 2 derived structure, it was apparent that the flexible regions are located within the loops, Residue-specific flexibility was observed from residue 25 to 35. Flexibility was also observed in another region from residue 55 to residue 59. The most flexible region was found from amino acid 117 to amino acid 223, for which the PLDT score was very low to low. However, the bone morphogenic protein 1 or BMP1 processing site is in this site, which is in between the amino acid residue QA and QE. The RMSF result agreed with the structural model, as the PLDT score of the AlphaFold 2 is an indicator of flexibility and disordered region. The mean RMSF was the lowest for AlphaFold 2 derived structure among the three structures, precluding the last 7 residues from 117 to 123. Next, we wanted to evaluate the intraprotein hydrogen bonds among the three structures. The presence of polar amino acids at the surface and non-polar amino acids at the core may support a higher number of intraprotein hydrogen bonds, and that also indicates the stability of the protein. Most frequently, loops of a protein are found near the surface of the protein, which exposes them to solvents and other proteins. This feature enables loops to play a crucial role in the protein structure, such as maintaining the protein structure, such as shielding the hydrophobic core or binding to other proteins and ligands. Among the three structures, AlphaFold 2 has the highest number of hydrogen bonds. So this figure represents the hydrogen bonds which are formed within the protein. So there are a total of 182 interprotein hydrogen bonds were found analyzing with Chimera, which is a molecular visualization software, and among them 152 hydrogen bonds were strong. The hydrogen bonds on the loop were longer compared to the hydrogen bonds which are within the beta sheets. The shorter, stronger hydrogen bonds make the core more compact, agreeing with the molecular dynamic simulation results. On the contrary, the hydrogen bonds on the loops and within the alpha helices are longer compared to the hydrogen bonds which are formed within the beta sheets. The compact core contained more hydrophobic amino acids compared to the loops as per the analysis of proteram. Next, we evaluated the solvent accessible surface area among the three structures. So among the three structures, the AlphaFold 2 derived structure had the lowest solvent accessible surface area, which may be due to the flexible loops shielding the hydrophobic core. However, further experimentation is required to validate this hypothesis. The radius of gyration is used to indicate the conformational transition and compactness of a protein. Among the three structures, AlphaFold 2 derived structure has the lowest radius of gyration. So we also compared the interprotein hydrogen bond and solvent accessible surface area, and we have observed an opposite trend. That means the higher number of hydrogen bonds is keeping the protein compact. As well as we also compared the average number of hydrogen bonds and the change of radius of gyration, and we have also observed an opposite trend among these two characteristics. So future directions. In future, this structure will help us to study interactions of this aminopropeptide domain with other collagens and with other glycosaminoglycans in the extracellular matrix. And next, we also plan to evaluate the in silico predicted interactions by biophysical, biochemical, and cell culture study. That's all from my presentation. Thank you.